Hello viewers, in my previous lecture on lipids, I had introduced you to complex lipids. And you know, like simple lipids, complex lipids are also esters of fatty acids and alcohols. But they also contain another group beside fatty acids and glycerol. So when we talk of simple lipids, we are talking of oils, fats and waxes. These are esters of glycerol and acids, higher fatty acids, that is they are triglycerides. And when we talk of complex lipids are also esters of fatty acids and glycerol. But besides fatty acids and glycerol, they also contain some other groups. And these other groups which are present in complex lipids may be phosphoric acid, nitrogenous base, carbohydrate moieties, proteins or sulfate groups. So depending upon what additional group we have, these complex lipids can be phospholipids, they can be glycolipids if the moiety is carbohydrates, then they can be lipoproteins if the moiety present there is a protein molecule or an amino acid molecule. So now let us continue the discussion further with phospholipids. Like what are phospholipids? As I've said, they are complex lipids and they will contain phosphoric acid and a nitrogenous base in addition to fatty acid and glycerol. So what we mean here is that when we are talking of a glycerol, when we are talking of an alcohol molecule in phospholipid, it may be a glycerol molecule or a sphingol or a sphingosine molecule. This molecule, that is the glycerol molecule or the sphingosine molecule, is the central molecule of phospholipids. And these then, we know that if we talk of phospholipids, that is glycerophospholipids, then in glycerophospholipids, glycerol forms diester with fatty acids. And these fatty acids may be polyunsaturated fatty acids or they may be just saturated fatty acids. And along with this, we have additional groups like phosphoric acid, nitrogenous bases, or we can also have a non-nitrogenous base. So now that means if I summarize like what is a glycerophospholipid, I would say a glycerophospholipid has two non-polar fatty acid chains. When we say two non-polar fatty acid chains, what we mean here is that we know that glycerol is a trihydric alcohol, that is the first hydroxyl group and the second hydroxyl group on glycerol will make a ester bond with fatty acids resulting in diesters. And then we have a phosphate group is attached to the third hydroxyl group by phosphoester group, and this phosphate group is also attached to polar amino alcohols. And these polar amino alcohols may be choline, ethanolamine or serine, as you can see on your screens. So now if we look into the structure of glycerophospholipids, which you can see on your screens, so as I've already told you, that glycerol is a trihydric alcohol. So the first alcoholic group of glycerol forms a ester bond with a fatty acid, resulting in an ester group here, and then it again reacts with a fatty acid giving us a ester group here. So we get two esters here, and the third OH group is attached to a phosphate group by phosphoester group linkage, and this phosphate group is then again attached to an alcohol group which can be amino alcohol, which can be ethanolamine or which can be serine, and through again phosphoester linkage. So this diagram which you're seeing on your screens is a schematic diagram. So if you are given this diagram, it should immediately come to your mind that this molecule or this diagram is representing a glycerophospholipid. And now we know these phospholipids are further classified according to alcohol present in them. They can be glycerophospholipids if the central molecule or the backbone of the phospholipid is glycerol. And they can be sphingophospholipids. They will be sphingophospholipids when the central backbone or the central molecule, backbone or the central molecule of the lipid will be sphingosine or sphingol alcohol. And you know that sphingol alcohol is an 18 carbon alcohol, whereas when we talk of glycerol, glycerol is a 3 carbon alcohol. So again, on your screens you can see the structure of glycerophospholipids. That is, the first OH of glycerol is attached to a fatty acid. Second OH of glycerol is also attached to a fatty acid, and the third OH is attached to a phosphate group, which in turn is attached to a amino alcohol. So now we can say that two fatty acids form ester bonds with the first and second hydroxyl group of glycerol. The third oxygen on glycerol is bonded to phosphoric acid through a phosphate ester bond. So if you see on your screens here now, I have shown you the structure here. So what we have is that in this color, this is the central backbone, or we can say this is the central molecule, which is the glycerol molecule. Now this glycerol molecule, the OH on the first carbon glycerol molecule undergoes reaction with a fatty acid, resulting in an ester bond here. Similarly, the second OH also reacts with a fatty acid, resulting in an ester bond. The third OH reacts with a phosphoric acid, resulting in a phosphoester bond, and then this phosphoric acid reacts, say, with choline, resulting in a phosphocholine ester bond here. So what we find in this structure, if you look carefully, this long chain fatty acids of esters form the non-polar part of the molecule, whereas the head, which is that of an amino alcohol, in this case what you're seeing here is choline, this is the polar part. So we can say that phospholipids are amphipathic in nature. Why are they amphipathic in nature? That is because of the polar bond, they are soluble in water, and because of this non-polar long hydrocarbon chains of fatty acids, this portion will be non-polar. This will be insoluble in water. Now this was the general structure of a phospholipid. Now, depending upon nitrogenous and non-nitrogenous moieties attached to glycerolipids, there are 7 types of lipids. First, the simplest phospholipid is phosphatidic acid. Next is phosphatidylcholine, phosphatidylethanolamine, phosphatidylserine, phosphatidylethanolamine, phosphatidylinositol, that is lipositol, and diphosphatidyl or cardiolipin. So phosphatidyl acid is the basic phospholipid, and all other phospholipids, that is other 6 phospholipids, are derivatives of phosphatidic acid. So now let us look into what is phosphatidic acid. This is the simplest glycerophospholipid. In this phosphatidic acid, a glycerol molecule is esterified with 2 fatty acids at C1 and C2, and the C3 is esterified with phosphoric acid. So as you can see on your screens, the schematic diagram for glycerophospholipid, that is the simplest one. So what we find in this case is that glycerol is linked to 2 fatty acids through 2 ester bonds, and then glycerol is linked to a phosphate group through phosphoester bond, and this structure is that of phosphatidic acid. Now what you have to remember is that if we replace the H of phosphoric acid with some nitrogenous base, it will give us derived phospholipids, and the first derived phospholipid that I would like to discuss with you is phosphatidylcholine, which is also known as lecithin. In lecithin, phosphatidic acid is linked to a nitrogenous base choline to form phosphatidylcholine. So what you have to remember is that when we are talking of choline or they're talking of lecithin, what you have to remember is that the basic unit is going to be the phosphatidic acid. And in phosphatidic acid, the phosphate group will form a phosphoester linkage with choline. So that is what it will give us the structure of lecithin. So if you look into this, on your screens, like phosphatidic acid, this is the glycerol is the basic backbone of cephalins and of this molecule. And in this, what we find, the first hydroxyl group and the second hydroxyl group are forming bond with fatty acids, and the third one is forming a phosphoester bond with a phosphate group. So this is the structure which if you recollect, is present in phosphatidic acid. So what has happened here, that there is additional phosphoester bond between the phosphate group and choline, which results in the formation of this molecule, that is phosphatidylcholine. And what you have to remember here is, in this molecule, the choline, this is the polar group. This is the head of the molecule and this is polar, whereas these two long hydrocarbon chains of fatty acids are non-polar. The next phospholipid that I would like to discuss with you are cephalins. Now, what are cephalins? In cephalins, the basic structure will be that of phosphatidic acid. Only the bases will change. So, if the base is ethanolamine or serine attached to phosphate group through phosphate ester bonds, then we get two cephalins, that is, first is phosphatidylethanolamine and the second is phosphatidylserine. So now if you look on your screens again, you can see the structure of cephalin phospholipid, that is ethanolamine. So in this part, this basic structure will be that of phosphatidic acid. That is, we have a glycerol moiety here. This is your glycerol moiety which is attached to two fatty acids, and the third one is attached to a phosphate group which is then attached to an ethanolamine. So if you look into this structure, so what we find here again, that this is the glycerol backbone which is attached to two fatty acid molecules, and then this is the head which is polar in nature, and these two are non-polar in nature. So now if we will discuss the next cephalin, which is serine cephalin, then what you will find is the only difference there would be the nature of the base present there. Base present in phosphatidylserine is serine, and this amino acid serine is linked to phosphatidic acid by phosphoester bond, and it will form phosphatidylserine. And this phosphatidylserine is important component of biological membranes. So as you can see on your screens now, what we find, that this is the glycerol molecule which is again forming the central backbone of the molecule. It is attached to two fatty acids through ester linkage, and the third OH is attached to a phosphate group by phosphoester linkage, and this phosphate group in turn is attached to serine, resulting in the formation of phosphatidylserine. Now you have to keep this in mind, that this part of the molecule is non-polar because it is composed of long chain fatty acids, that is the long chain hydrocarbons, and we know hydrocarbons are non-polar in nature, whereas this part of the molecule is polar in nature because of the presence of negative charge on oxygen. If it is an ammonium salt, we will have a positive charge here. So it is polar in nature. So what you have to remember is that heads are polar when we are talking of cephalins or lecithins, and tails are non-polar. So lecithins and cephalins are abundant in brain and nerve tissue. But what is the difference between the two? We can differentiate between the two only on the basis of the bases, nitrogenous bases present in them. In lecithin, the nitrogenous base is choline. Whereas when we talk of cephalin, then in cephalin, the nitrogenous base may be ethanolamine or serine. And if you compare the two structures, the other two structures are similar. That is, we have a glycerol backbone, here glycerol backbone, and this glycerol backbone is attached to two fatty acids in both the structures, and the third one is attached to a phosphate group. The OH is attached to a phosphate group. So this residue here is common in both. So where do they differ? They differ in the bases which are present in them. So on the basis of the bases which are present, you can differentiate between cephalins and lecithin, right? Now we go on to a simple structure. Now I just want to quickly recapitulate what we've done till now, that this is a simplest glycerophospholipid. That is, this is your phosphatidic acid. Now this is the basic molecule which is going to be present in all the phospholipids, glycerophospholipids. Where will it differ? It will differ in this group X. If this group X is hydrogen, then you have to remember that that molecule is phosphatidic acid. And if this group X is ethanolamine, then you have to remember that it is phosphatidyl ethanolamine. If it is choline, that is this small structure, this base is present, then it is phosphatidyl choline. And if we have serine present, then the molecule is phosphatidyl serine. So you do not have to mug up the structures, you just have to remember what is the basic structure of phosphatidic acid, and then what they ask you, whether they ask you in phosphatidic ethylamine, phosphatidyl choline or phosphatidyl serine, you will change the bases and you will get the desired glycerophospholipids. Now another glycerophospholipid which is very important and which is a non-nitrogenous phospholipid, it is lipositol. Lipositol is made up of inositol. In it, the carbohydrate which is present is a inositol. So what is lipositol? Lipositol is a carbohydrate derivative. So we do not have a nitrogenous bases like serine, ethanolamine or in it. What do we have in it? We have a carbohydrate moiety in it. And therefore this will belong to a category of glyceroglycolipids. So in glyceroglycolipids, the base present is myoinositol. And myoinositol, please remember, is a carbocyclic sugar molecule. So if you see on your screens, what you find that this is a molecule of inositol, and this molecule of inositol is attached to the phosphatidic acid. This is the basic unit which is present in phosphatidic acid, and this unit is known as phosphatidyl because we've removed hydrogen from here and we are forming a phosphoester bond between phosphate and inositol. So this molecule is known as phosphatidyl inositol. Now what are the uses of phosphatidyl inositol? It is a very important, please remember, it is a glycolipid, glycoglycerolipid. It is not a phosphoglycerolipid, and it is present in soya bean and in brain and spinal cord of animals. And it is this which is responsible for action of sex hormones, oxytocin and vasopressin. So that means now if we look into this, again, I have made a table for you, and in this you can see that if we are replacing the R with inositol, we are getting lipositol, and if we are replacing this H say with phosphoethanolamine, we are getting acetylene, and if we are replacing it with serine or choline, we are getting phosphatidylcholine and phosphatidylserine, which are examples of lecithin. Now, next very important phospholipid is diphosphatidylglycerine. It is also known as cardiolipin. Why is it known as cardiolipin? Because it was first isolated from cardiac muscles of calf, and hence the name cardiolipin. And it has a very unique structure. What is unique about it? What is unique about this structure? That it consists of two phosphate residues. That is, it consists of two phosphatidyl residues, and it has 4 kind of fatty acid chains. So now that means that 2 molecules of phosphatidic acid are linked to 1 molecule of glycerol. So if you look on your screens here, what you're seeing on your screens is the structure here. So what we find that this molecule here is 1 phosphatidyl group, and this group here is the second phosphatidyl group here, and this is the central backbone, which is that of glycerol. So what we find in this case is that the first OH of the glycerol is attached to the phosphatidyl unit moiety, and then the third OH forms a phosphoester bond with phosphatidyl moiety, resulting in 2 phosphatidyl groups here. So therefore this lipid is also known as diphosphatidylglycerol. And what we find here in phosphatidylglycerol, how would you write the structure? See, for what you can do is that in first case this is written simply here. In this case you don't have to do much, but supposing if we were rotating here, what you can do is that in first case you take this molecule as such, that is 1, 2 and 3, and second case what you do, you flip the molecule, and when you flip the molecule, what you will find is that the phosphate group of the second moiety will come in front of the third carbon, and it will get 2 phosphoester bonds in it with phosphate moiety, that is with phosphatidyl group here and with a phosphatidyl group here. So this is a very different glycerolipid when it is compared to other ones. Now, what are this importance of cardiolipins? Cardiolipins are present exclusively in the inner mitochondrial membrane, and supposing with all the lipids which are present in mitochondrial membrane, 20% of them are cardiolipins. And why are cardiolipins important? Because they are important for optical functioning of mitochondrial enzymes involved in enzyme metabolism. Now in my next lecture, I shall be discussing with you ether phospholipids, and then maybe we will continue with the sphingolipids. For this lecture, I had consulted the books by Bruce, McMurray-Jones, Finar, Morrison and Boyd, and Solomon. Thank you.